Okay, I'm sorry. My computer decided to have a little hissy fit. So I had to split that into two parts. Anyway, so this is part the second of our prokaryote discussion. So I was talking about organization within prokaryote cells. The one thing I should emphasize, certainly eukaryote cells are much more elaborate in their organization. But remember that the prokaryote cell has to do all the same things that a eukaryote cell does. It performs almost all of the same metabolic functions. It has to replicate its DNA. It has to transcribe its DNA translated. It has to do all of the things that a eukaryote cell does. But it has to do it within the bounds of a cytoplasm that lacks membrane-bound specialized areas to perform those functions. When we look at the processes that are taking place inside bacterial cells, what is remarkable about them is often their similarity to the processes that we see in the eukaryotes, which we regard as being so much more advanced. We could probably think of eukaryotes as being much more advanced in their organization rather than in their biochemistry, because prokaryotes are in fact very advanced in their biochemistry. They are that is clear from their diversity and from their success. When we look at a process like DNA replication, transcription, etc., we realize that, in fact, there's very few differences between the prokaryotes and the eukaryotes, but there are some. There are a few differences, slight differences. And those slight differences are important in terms of treating disease, for example, because they give us the opportunity of using antibiotics, which will interfere in those minor differences, using those minor differences as their leverage, so that when we use an antibiotic, it operates against prokaryotes and not against eukaryotes, because their processes are slightly different. Now, the organization of the genome in the prokaryotes, as I've said, the major part of the genome is a large circular chromosome which lies inside the cytoplasm, unbound by a membrane. But there may be other pieces of DNA as well, and these are usually in the form of circles, little tiny circlets, which are called the plasmids. And plasmids are often contain information which is useful to the cell. Often, for example, they may contain genes which confer resistance to antibiotics. In some cells, they confer pathogenic ability. And the really significant thing about plasmids is that they are actually infectious. They can be passed from cell to cell, from organism to organism, even across specific and generic lines. And for example, a cell which bursts in the environment, which dies and bursts, may release its plasmids into the environment where they can be taken up by other cells. So this is an important way of transferring genetic information through a population. This is one of the things which confers such success on the prokaryotes. We recognize these particular things. First of all, their rapid reproductive rates, their ability to form very large populations, exploit an environment very, very quickly. Second of all, they mutate very quickly. And thirdly is what I've just mentioned. They are able to actually transfer genetic information from cell to cell. They're able to transfer genetic information, we say, horizontally through the population. And they can transfer this information in various ways. Now, their principal mechanism of reproduction, of course, is asexual. It's by binary fission. It's essentially a form of mitosis, because what the cell is doing is it's duplicating its genome. By the way, the plasmids also duplicate themselves. And the cell then divides in two by pinching the cytoplasmic membrane together to divide the two cells, one from the other. But these cells are mutable. Bacterial populations, although mutation rate, when we look at it, may be low in an individual cell, when we look at a whole population, the population growth is so tremendous that mutations appear with regularity in bacterial populations. And once an advantageous mutation has evolved in a population, it very rapidly accumulates because the populations reproduce so quickly. That's the first thing. And second of all, they're able to pass these advantageous genes from cell to cell. And the way in which they do this by 3 different mechanisms. The first I've already mentioned. That is that they can, that plasmids released into the environment are actually infectious and can be taken up by other bacterial cells. This process is called transformation. The second way in which new genetic information is acquired is actually through viruses. Now, this may sound a bit strange, but think about this. I told you that viruses, when they reproduce, first they get viral particles, viral proteins assemble themselves inside bacterial cells, and they then supposedly package the viral genome into the capsid, into the new capsid. Sometimes those viral particles actually don't package viral DNA, for example. Sometimes by accident they package bacterial DNA. Remember that in a lytic situation, the bacterial DNA is chopped up into small pieces. Some of that DNA may get packaged into a viral particle. And if that viral particle then infects another bacterium, what they do is they inject the genes from another bacterium. And those genes may be useful. They may confer an advantage to the cell that has just been infected. So that is a process called transduction. So contrast transformation and transduction. The third way in which bacterial cells exchange genetic information is through a process called conjugation. Now, conjugation means coming together. And that is exactly what it is. If you want to think about it this way, it's almost like bacterial sex, in that they are exchanging genetic information, accepting that the exchange is a one-way exchange. The way it works is that we have a cell, say this one here, and this cell has information which it enables it to build a special protein tube called a sex pilus. It builds that sex pilus and it contacts another cell. This cell does not have that ability. And the sex pilus pulls the two cells together like this. And this cell then copies off a section of DNA with certain genetic information on it, including the ability to make a sex pilus. So the exchange is one way. We have this cell here with information, with genetic information that it is going to confer onto this cell here. This is the process of conjugation. And you'll realize that we often call this an F plus cell here, and this an F minus. And the F plus cell converts this cell into an F plus. This cell can now go on to convert other cells. And the information that is conferred is often the same kind of information that you would find on a plasmid, often resistance information, pathogenic information, all of it is something that confers resistance to a mineral, to a metal or something like that. Very often useful information. So all of these things, the transformation, transduction, and conjugation, we call horizontal transfer of genetic information. Vertical transfer is information which is conferred from one cell to its daughter cells when it divides. This is information which is coming in from other cells, and it is conferred horizontally. Okay, so all of these things have led to bacterial populations, prokaryote populations rather, becoming incredibly diverse. And the one way in which we see this diversity is in their nutrition, is in the way in which they nourish themselves. First of all, there are lots of autotrophic prokaryotes. An autotroph is something which takes carbon dioxide and water and some form of energy and puts those simple molecules together to form sugars. So that the most familiar process for us is photosynthesis. But the process of photosynthesis evolved in the prokaryotes, and it is not the only autotrophic process. So photosynthesis is a photoautotrophic system. That is, it uses light as the source of energy to put these compounds together to form the sugars. But there are many bacteria, many prokaryotes rather, which are able to use carbon dioxide and water and put them together using energy extracted from a chemical compound, such as hydrogen sulfide, for example, and various others as well. It's using the same system, the autotrophic system, CO2 plus water, but you need energy to join those together to form the carbohydrate molecule. The energy source is what distinguishes the photoautotroph from the chemoautotroph. The majority of prokaryotes, however, are heterotrophs. And what they do is they are using an already created organic molecule as their source of molecules to build their own biomolecules. But they also need a source of energy. And there is a group of prokaryotes which derive their molecules from organic molecules, but don't derive their energy from those molecules. Instead, they derive their energy from light. And those we call the photoheterotrophs. Very, very unusual and very strange way of life. But they simply use organic molecules as a source of molecules, not as a source of energy. Most prokaryotes, and in fact all eukaryotes, are chemoheterotrophs. That is, that they use already created organic molecules as a source, both of molecules for biomolecules and as their source of energy. And those we refer to as chemoheterotrophs. We are chemoheterotrophs. So to make sure that you understand these four distinctions: phototrophs, chemotrophs, autotrophs, and heterotrophs, and how we actually put them together. Here's a little question for you to answer. Okay, so we realize when we look at that tree of life, we realize that everything that we see living today evolved from a common ancestor. And that was about between 3.5 and 3.9 billion years ago that that ancestor actually evolved. From that common ancestor have evolved all of the domains that we saw previously in the tree of life. And that has been an extraordinary series of evolutionary events. But when we look at the prokaryotes, we look at their structure, and we look at their functioning, we see all of the roots of the structure and functioning of every other living thing on the planet. Here, this, just to remind you, is the divisions into the three domains: bacteria, archaea, and eukarya. Here, and these should by now be pretty familiar to you. We've covered most of these, and I wouldn't worry about the membrane structures here, or the RNA polymerase. The only one I have told you about before, or sorry, I told my class, but you should have heard about before. The one other difference that we notice between the bacteria and the others is in bacteria, during translation, the very first amino acid that is included in a polypeptide is formyl methionine. In both the archaea and the eukarya, it is methionine. So apart from this, you should please remember all of these differences between the three domains. Okay, so bacteria, when we just look at them, we see a huge amount of diversity. This is one of my favorites. This is called Thiomargarita namibiensis. This is the largest of all of the bacteria. And if you think that most bacteria are around between 5 and 10 microns long, this is Thiomargarita, is 200 microns long, which makes it big enough to see with the naked eye. It's a quite extraordinary bacterium that lives under very specialized marine circumstances, but nonetheless, there it is there. And by the way, this is a chemoautotroph. This is one of the very smallest of the bacteria. This is chlamydia, and this causes sexually transmitted infection, but also causes an eye disease called trachoma. And it is very small, very reduced bacterium, and it's an obligate intracellular parasite. These are spirochetes. I should explain to you that the spirochetes are spiral, but you see how it's a very tight spiral. And that is because it's wrapped around a flagellum that runs down the center here. And it gives the spirochetes a unique ability, and that is that they move in a particular way. They spin on their own axis. And they are the only bacteria which are able to move forward and to move backwards. These are cyanobacteria, and cyanobacteria are the bacteria in which the process of photosynthesis, photoautotrophy evolved. And the cyanobacteria still contribute about 20% of the world's atmospheric oxygen, particularly marine cyanobacteria. These are gram-positive bacteria. And this is one, if you've ever had strep throat, this is the beast caused your strep throat. And you can see it's a chain of small rods all joined together. The archaea, unfortunately, we have little to say about. They do share some traits with the bacteria in that they are prokaryote. They have a circular chromosome, but they have a unique cell wall. It does not consist of peptidoglycan, and there are some differences in the structure of the membranes and various other biochemical things which differ from the bacteria, which are more similar to the eukaryotes. And as I told you before, when we look at their phylogeny, we realize that the eukaryotes and the archaea are closer together than the archaea are to the bacteria. So they are more similar to the eukaryotes. And this is our eukaryote cell probably arose from an archaeal-like ancestor. And that is the reason for our sisterhood, if you like. They're quite extraordinary organisms, often live in very extreme environments. They can live in very, very high salt environments without dehydrating, which would happen to a normal bacterial cell put into a high salt environment. Remember, normally in a normal marine or aquatic environment, water floods into a bacterial cell. Put it into a high salt environment and the opposite happens. Water leaves the bacterial cell and it shrivels, pulls away from the cell wall in a process called plasmolysis, which you probably saw in plant cells in your lab last semester. It's the same process. The cell withers, as it dehydrates and pulls away from the cell wall. Very, very dangerous. Very few bacteria are adapted to withstand that kind of, but the archaea are. And these may occur in in the Dead Sea, for example, in areas like this with extraordinarily high salt content, they can still exist. They are also extreme thermophiles, and they have to have their DNA and their proteins adapted to withstand high temperatures. And at deep sea hydrothermal vents, for example, the water may be well over 100 degrees Celsius, and yet they can still survive. Oh, here's a little tree just to remind you of the position of the archaea and of the eukarya relative to the bacteria. So this singular importance of the prokaryotes is the absolutely crucial role that they play in the bioecology, and especially in recycling of chemical elements between living and non-living components of the biosphere. So I remind you of something, and that is that essentially our planet is an enclosed unit. It is a closed system for molecules, not for energy, but for molecules. The molecules we have present on the planet is what we have to deal with. And what living things do is they use energy to build molecules from components that they extract from the environment. So they are living things are continually building molecules. If those molecules never decayed to their smaller components, the molecules would be used up. They would end up being stored in these vast stores of inert biomolecules, and the whole of life would grind to a halt. But that's not what happens. The lifespan of a biomolecule is usually relatively short. The duration of the lifespan of the organism that built it, for example, maybe some time afterwards, but it is the role of, especially of the prokaryotes, to break down those molecules into smaller components. And they do so by releasing enzymes into the environment, which break down biomolecules into small enough components that they can be absorbed by the prokaryotes. But their absorption is relatively inefficient. And as a result, they are responsible for breaking down a huge amount of organic matter and making that available to other living organisms. In addition to that, there are elements such as nitrogen are extremely non-reactive and yet are essential parts of biomolecules. For example, we have to have nitrogen in our proteins. It's an essential component of our proteins. So we have to have a continuous source of nitrogen, but nitrogen is extremely non-reactive. We breathe in nitrogen the whole time, but we can't make any use of it. We can't take that nitrogen and put it into proteins. Instead, we depend upon prokaryotes to take atmospheric nitrogen and convert it into a form which can be used organically. That is usually done in association with plants and in the soil, especially by groups of prokaryotes, which are able to take nitrogen and convert it largely into ammonia initially, and that ammonium is then used metabolically to construct other nitrogenous compounds. Though that nitrogen fixation reaction, as I said, is often done in symbiosis with plants. And this is the last of the really important features of prokaryotes, is that they often establish elaborate associations with other organisms, especially with the eukaryotes. That is just one, the nitrogen fixation is just one of those. We have our own bacterial associations, which are equally important to us. And for example, we have an extensive bacterial flora which is associated with us on virtually every mucous membrane that we possess, for example, there is a layer of bacteria which are completely natural, completely normal. The only place where this is not true is deep in the lungs, which are kept not entirely sterile, but more or less sterile. But in our noses, in our throats, throughout our guts, and in our reproductive system, we have resident bacteria which are completely natural and which form part of our normal bioflora, our normal microflora. The particularly important is the microflora of the gut, which is extraordinarily diverse. We have thousands of species of bacteria in our gut, and they perform all sorts of useful functions for us. And we're discovering that they're in fact involved in many aspects of our health, which it's actually quite difficult to understand how they are involved, but they're even involved in mental. It appears they're even involved in mental health. It is true, of course, that bacteria can cause disease in human beings as well. And almost half of the bacterial of the diseases which humans suffer from are bacterial in origin. Some of them have plagued human beings for centuries and continue to do so. The disease of tuberculosis, for example, caused by an organism called Mycobacterium tuberculosis, kills close to 2 million people a year, and actually the number is increasing, not decreasing. That is a typical disease which is transmitted from person to person by contagion. Other bacterial diseases are transmitted by vectors such as insects, and the one that you receives yearly publicity in the United States is this disease, Lyme disease. This is Lyme disease transmitted by a tick, and you can just see the little tick there. It's actually a spirochete, shown there, and this is this first symptom of Lyme disease. It creates what is called a bullseye rash. There it is there. And that's a typical vector-borne bacterial disease. There are many, many, many others. For almost the entire history of mankind, these bacterial diseases have caused mass mortalities. That is especially true of diseases which are transmitted by water and by food, which cause guts and diarrheal diseases. These are still extremely important, especially in children, kill millions of children every year. In addition, there are all sorts of other infection, all of these sorts of things. In the 1940s, when antibiotics were first discovered and invented and then commercialized, we thought we'd entered a golden age where all bacterial diseases would be curable. And for a time, at least, that seemed to be true. Antibiotics were enormously, enormously successful. But unfortunately, we need to remember what we just learned about the success of bacteria in being able to reproduce very rapidly, mutate, and acquire advantageous genetic structure very, very rapidly. Presented with an environmental threat such as antibiotics, bacteria respond in exactly that way. And they begin to develop resistance. And resistance can develop incredibly quickly after the introduction of antibiotics. We've reached a stage now, the first true antibiotics that we used were the penicillins. They were enormously successful, especially in eradicating infectious diseases of the gut and infections of wounds and this sort of thing, which formerly had been extremely high mortality. But very, very soon after the introduction here, they were introduced in the 1930s, They were discovered in the United States. They were only introduced around about here towards the end of the Second World War. But really, by the late 1940s already, it was noticed that certain bacterial strains were becoming resistant. The response is then to go out and look for new antibiotics, which is exactly what we do. And some of them extremely powerful, but the more powerful they get, the more likely it is that they also have side effects which affect the people that are receiving them. But it doesn't matter which antibiotic, no matter how powerful it is, bacteria always seem to find a way through. We're now reaching a critical stage where the development of these resistances in prokaryotes is far outstripping our research ability to find new antibiotics. Most of these antibiotics are derived from living things, from fungi and other bacteria. And the research simply can't keep pace with the rate at which resistances are developing. It is an extremely serious situation, and deaths due to bacterial disease are actually on the increase because of the occurrence of these antibiotic resistances. But we need to remember that bacteria are very useful to us as well. We utilize bacteria under many different circumstances. So, for example, all your fermented products, yogurt, cheese, all of those things, those are all produced by bacteria. We utilize a system whereby we encourage the growth of harmless bacteria to such an extent that their population creates conditions which exclude potential pathogens or spoilage organisms. That's exactly what we do when we make yogurt, or we make cheese. We use it in even sausages like pepperoni. They are actually fermented. That is their preservation method. All of those things like sauerkraut, that is fermented cabbage. These were, in human populations, were developed as ways to preserve food, especially to preserve it over winter time, so that you have a source of nutrition. You may not know what ensilage is. Ensilage is like sauerkraut for cattle. It's a fermented lucerne or fermented grass. And again, it was fermented by bacteria. It's fermented by bacteria. It's still made. Fermented by bacteria, and it forms a product which can be kept over winter or for a long time to feed cattle. Soy sauce, all of these sorts of things. These are all fermentation products, all from the controlled use of bacteria. And in addition to that, we need to remember that there is now a whole industry of biotechnology using bacteria industrially to produce useful products for human beings. Everything from pharmaceuticals to oils and solvents and all sorts of things. Okay, so that very, very quick run through viruses and prokaryotes. I'm sorry I had to split this into two, but excuse me. I will see you on Monday.